So I'll be sharing some work that my group has been doing over the past few years in computational pathology. Here are my disclosures. And this is just a list of everything I'll be talking about today in case you want to go back and look at some of these studies later. So my group predominantly focuses on computational pathology. So when glass slides are digitized, they look something like this. So large images that are hierarchical and harbor information at many different scales. They're more like satellite images, something similar to what you would see on Google Maps or Google Earth. These slides harbor a lot of information. The interesting thing about pathology is that it's one of the oldest diagnostic modalities, but it's also one of the oldest research modalities. So it's been around for a very long time. As everyone here knows, it's used for very fundamental biological research, but on human, animal, all kinds of tissue, but also for clinical diagnostics, right? So, but a caveat is that this is, despite being one of the oldest modalities, is also a modality that's only recently begun to be digitized. I think the first scanner came about 20 years ago. And still, most hospitals in the country are not entirely digital, and the standard of care remains looking at glass slides under a microscope. All the other modalities that came about later, like genomics and other kinds of information that we can extract from tissue, are by default digital and computational. So we still haven't incorporated information from this modality into our diagnostic, prognostic, therapeutic response, or resistance prediction paradigms. So what we predominantly do is that we essentially try to analyze these images using computational machine learning tools to get to everything in the red box here, whether it's early diagnosis, prognosis, response to treatment, patient stratification, and so forth. Essentially discovery from this kind of data. But we also incorporate information from other modalities where necessary to try to fulfill some of those objectives. So I'll be talking mostly about the work that my lab does in human pathology, but we also do a lot of work in preclinical drug screening and toxicology and how to make those processes more efficient. And a lot of those questions essentially require the same computational tools that we build, but I won't be talking a lot about that aspect today. So the history of the field is very interesting, and I always like to talk about it. So Judith Previtt was a real pioneer in this field, and she's really an unsung hero because some of the work that she was doing in the 60s and 70s is phenomenal around how she was thinking about this problem. She worked at the University of Pennsylvania, then at the NIH, and finally at the University of Uppsala, where she had access to large mainframe computers. But this study from 1963, and in particular this study from 1965, if you look at how she was analyzing these images, so she realized that these images are very large. She was chopping them up into smaller patches. We still do this today because we can't fit these mega images into GPU memory, at least not to do the kind of training we do. And was storing these in magnetic tape using optical density, and then using very fundamental line profiles to assess whether an image is deemed to be positive or negative. What a lot of people don't know is that this became the basis of the very first FDA-approved pathology algorithm. It was called AutoPap for pap smears in the 1990s, and has been used since to reduce workload, to triage out negative cases, and is one of the only pathology-based algorithms that is approved for primary diagnosis, such that the pathologist does not have to go back and look at that particular image. And then, with in more recent times, there's been a lot of work using handcrafted features, essentially reflecting how we perceive these images, and coming up with features that would be reflective of those of that perception. And then post 2015, the utility of convolutional neural networks, and this was predominantly achieved by pixel level annotation on these slides. However, this is very far removed from how this data is available and collected clinically. So clinically, we don't collect any pixel level annotations on these images. Once you have these annotations, you can separate that data into buckets and use very fundamental deep learning algorithms to classify cancer, normal, subtype, grade, anything. But the issue is the labels. We don't have pixel level annotations as part of the regular clinical workflow. And that's where all these weakly supervised methods that have been developed for this kind of data come in. And the two most common ones are the utility of using graph convolutional neural networks, where you can segment all the cells and build a cell graph on top of it. That can be quite computationally complex, especially given that each case can have a multitude of slides. The average slides per case are between 8 to 9, but a single case can have up to 100 gigapixel slides. So the most common approach, which comes to mind from a machine learning point of view, is the utility of multiple instance learning, right? Because you have large images, maybe you can split them into smaller patches, and per case you also have a multitude of images. So whenever you have lots of instances, you can use multiple instance learning, assuming that a slide is a bag of many small patches. And if you're using a max pooling based aggregation function, the assumption would be that even if one of those patches is positive, the entire slide would be considered to be positive. An issue with this approach is that this makes this setup inherently data inefficient, because the weights and biases of the model could be updated using a single 256 by 256 patch. And this is what was articulated in this 2019 really seminal study. It was published at the Memorial Sloan Kettering Cancer Center, where they showed that you needed about 10,000 whole slide images to get the same level of performance you would get with slide level labels. This is back in 2019, and they're focusing on targeting very fundamental problems like detection, subtyping, grading, and prognostication, right? But we really wanted to see if there's a more data efficient way to do this, and we started working in this area around the same time in 2019. So we can target the more interesting problems in pathology, like rare cancers, clinical trials, patient stratification. So in 2021, we published this study, and we called the method clustering constrained attention MIL. And it used a number of different bells and whistles that were developed for computer vision. We brought them into pathology, and this included attention-based multiple instance learning, the utility of pre-trained encoders, and using clustering as a mechanism to cluster together similar morphologic regions within the slide. So a slide would have lots of normal regions, lots of disease-positive tumor regions. So clustering can potentially help associate similar regions together across entire cohorts. And we made this tool publicly available, and over time this has sort of become very widely used and has been applied to almost every disease model that's tackled using for human pathology. So the basic workflow is actually very simple. It's you have whole slide images. You scan them. Once they're scanned, you have a whole slide. You have a large hierarchical image. You can segment the tissue, patch it into smaller patches, and then you can extract features, right? So there was a lot of work that was training this end to end, but that can be inherently slow, computationally limiting. But if you use a pre-trained encoder, in the beginning we were just using a ResNet that was trained on ImageNet features. So standard between 2019 and up until recently, when we started using self-supervised learning, it was really just a ResNet that was trained on ImageNet features. You have eventually get to patch embedding. There's some aggregation, and then the slide level embedding gets fed into a predictor. And you can pose this as being any kind of machine learning problem. This could be a classification problem targeting disease detection, subtyping, grading, or it could be a prognostication problem where you're essentially solving a ranking problem, ranking patients into distinct risk groups, or it could be treatment response, anything that you practically have a label available for. So a number of different applications we targeted back in, we started this in 2019. I'll just say a little bit about those applications. So we looked into cancers of unknown primary. When I think most people here are familiar, when cancer metastasizes, it often becomes unclear what the origin of the tumor might be, and most treatments are based off of the origin. So this is a critical problem. These patients typically undergo large workups of not only diagnostic pathology, but also radiology, endoscopy, repeated molecular testing to try to see if the origin can be determined. And if an origin is not determined, the patient often cannot enroll in clinical trials either. So in this study from 2021, the primary question we tried to answer is that, can we use conventional H&E images to determine what the origin might be? And we believe this would be possible because there's lots of studies showing that you can predict molecular alterations, or essentially proving that there are more morphologic correlates to molecular alterations within the histology image. And we showed that you could predict what the origin is, effectively solving first the binary problem, whether the case is primary versus metastatic, and then an 18-class classification problem about what the origin might be. And then looking at the top 3, top 5 predictions was critical, because it can give you an essential hint for what the origin might be, and you can order ancillary tests like IHCs to confirm that origin. That reduces time, and this is something that we have implemented at MGH. That for every metastatic case, we're able to look at what the possible origins might be, and then already order those ancillary tests before a physician starts looking at it, before a pathologist starts looking at it. More recently, we have also used all the molecular data that we had available, that was available to us from the Dana-Farber profile cohort, and we have tried to combine histology and molecular data using very fundamental form of fusion. This was a study that really started a while ago, but then the amount of validation that was needed to do this took several years, because we needed to get data from other institutions, and it's very difficult to get cancers of unknown primary cases where the origin was already determined. But this will be coming out in the next couple of weeks, where we try to combine histology and molecular data to see if we can improve origin prediction. And we basically assess that how well can you predict the origin just from molecular data, from histology, and what happens when you combine histology and molecular data. Another application we targeted using the same sort of paradigm was around endomyocardial biopsy assessments after heart transplants. Patients often undergo repeated endomyocardial biopsies or heart biopsies to determine if the donor heart is being rejected by the recipient. This is a problem where there's a large intra and intra-observer variability, and we wanted to see if it's possible for us to standardize this a little bit more using these machine learning techniques. This was a study from back in 2022. I mean, I wouldn't go into the details, but there's a lot of analysis around what happens when when you have human in the loop assessments for these biopsies. So another sort of application was trying to see if we can combine histology and molecular data to improve prognostic models. So we know that molecular data is directly very prognostic. There are a number of these commercially available prognostic assays like Prosigna, Oncotype DX. All of them are they're good to a degree. They're imperfect, but they do work. So, but we basically wanted to answer 2 questions. Is it possible for us to combine histology data with the molecular information to see if we can improve prognostication? Because histology data is relatively free, right? It's always there, and it's available. It's a very cheap modality. So if you can prognosticate patients by combining it with molecular data, why not? And the other question would be that, can we predict similar patient separation just from the histology alone? And the third one was effectively around looking at, once we have made these predictions, they're all done in a weakly supervised manner, then can we go back and look at what's important in the molecular profile, what's important in the morphologic profile, in an attempt to make assessments, discover new markers, and we did a lot of analysis on the 14 different cancer types that were assessed, and we have since made all of that publicly available, and people have found all kinds of morphologic features that are consistent across patients. This was a Cancer Cell article from back in 2022. So coming back to the ML paradigm that we're effectively using, so it became clear that the feature extraction component is the most important component to me. Started experimenting with self-supervised learning as it was becoming popular initially by using contrastive predictive coding. Back in 2021, it was a NeurIPS article where we showed that you can use contrastive predictive coding to improve feature representation for these large multiple instance learning problems. In 2022, we developed something that we call hierarchical image pyramid transformer that makes use of the inherent hierarchy in the pathology image, and uses self-supervised learning on top of it to improve representation at various different hierarchical scales. That's a CVPR study from 2022. And then last year, we published these 2 foundation models. And I think that the critical inflection point was that when Meta showed in their DynoV2 study that the diversity of data is much more important than the quantity of data, something that we always knew, but it was like a proof. That gave us an opening that we don't effectively need to scan every single slide through our history to develop a foundation model for pathology. We could potentially go back our archive and look at what are the most diverse cases, pull those the most diverse cases and scan those, and potentially that would be good enough to develop a foundation model representing human pathology. So to test that hypothesis, we basically did exactly that. We looked through Brigham and MGH archives, looking at all the pathology reports, finding what are the most unique cases, and pull those cases, scan them, and then develop these models. The other one was a vision language model, and these models were made publicly available and have since been downloaded over 1.4 million times, and they've been out for about a year, and we're still investigating what the impact has been, but they've already been used in over 400 different studies. And I think one key aspect is that pathology data is so fundamental to everything we do in biomedicine in general, but there's never been sort of a clear way on how to incorporate this data. It's large, complex, all of that, right? So it exists, but if you can have lower dimensional representations of that data, it's quite easy to incorporate that into any question that you might be asking.
Sorry, could you define what you mean when you say like looking for the most unique pathology images? Like, is that defined by a pathologist, or is there some sort of metric for diversity?
We looked at a number of different ways. I mean, you could just embed all the pathology reports and see in feature space how they separate. That's what we initially did, because we have 22 million pathology reports. So how do you figure out? So we started by looking at all the pathology reports, then embedding them, and then.
So the reports are images and text, or just images?
Just text.
Just text. Okay.
Reports are just text.
Okay.
No image. So they typically. So there's a lot of diversity across the reports. They have diagnostic information, grade information, clinical correlations, and it varies a lot across pathologists. So pathologists in community hospitals would often write a detailed report with morphologic description and everything in there leading up to the diagnosis. A lot of pathologists in more advanced centers would just say, well, this is renal cell carcinoma, full stop, and then, right? So there's culture, but they would always have information about diagnosis, if there's any prognostic indication, such as grade. So there's some standardization to it. And then there's also synoptic reports. So we didn't look at synoptic reports. We just looked at the descriptive reports, embedded them, and looking at it in feature space. That was one way. But we also looked at, just by looking at the diagnosis, how diverse it is. And then there was some existing data that we had collected in the past for other projects that was also used. And in often cases, some of the data that we were collecting was very diverse to begin with, because we're dealing with rare disease and other kinds of indications. Yeah. So in this, so it was around 100,000 cases. We have 100 million pathology images from 100,000 cases. This was the initial version of the model, and it used the DynoV2 framework for training. And at the time, we compared it with a number of other models, as well as just the baseline ResNet 50 trained on ImageNet. That was the most commonly used encoder for pathology images, and it was applied to 33 clinical tasks downstream. The radar plot is a terrible way to look at it, but it does capture all the different tasks that were done and what the comparative analysis is with other kinds of models, including looking at classification, retrieval, or image search, segmentation, and both at origin level and at a slide level. So these are just some results for slide level classification, and it has very difficult tasks and also relatively easy tasks, and we can see what the performance difference is, for example, with the ResNet baseline, which was typically used before we put these studies out. There's been a lot of foundation models since, and then the performance of these models is beginning to saturate, but it's important to also see that few-shot performance has improved significantly over the years. I mean, back in 2019, we're looking at using 10,000 slides to try to capture performance that one would get with pixel level annotations, all the way up to more recently, where you can use a few slides in a few shot manner and still get clinically useful performance. So in parallel, we also worked on this study where we tried to see if we can contrast with text to improve representation. I think this study sort of started this basically observing everything that's going on in machine learning, where we're seeing that contrasting across data multiple kinds of data has the capability of improved representation. And this is great for pathology because there's a lot of morphologic description that's available for pathology. And these kind of contrastive models make use of the redundancy across modalities to improve representation for each individual modality. In this case, this was more of a proof of concept where we used 1.1 million image caption pairs from the PubMed open access database. I mean, a lot of people have asked us, why have we not used the entire PubMed database? There's no way for us to do that. There are licensing issues, and that data is also not, just readily available. So it was limited to the PubMed open access database, and we sort of just used human tissue and then used morphologic description that is corresponding the image. And by contrasting between the two, this is a distribution for where all the organ systems that were used. We showed that you could improve representation, and there's a lot of analysis around how this compares to image only. This example is for few shot learning, and we can already begin to get clinically useful performance using about eight or nine slides. There's been quite a lot of independent validation of these models. Here's some validation that came from Jacob Cather's group in Dresden, and there's also some validation from Thomas Fuchs's group at the Memorial Sloan Kettering Cancer Center and from Mount Sinai, where they've used this on their own data. And I think that there's lots of models around for pathology now, lots of different foundation models. They've all kind of saturated in performance, but one key aspect is that because we really focused on diversifying the data, the models continue to be state of the art. And we have put out additional models that use more data and have also answered some fundamental questions around how to build these models in an effective manner. So the next obvious step for us was to see how can we get to a slide level representation. So Uni and Conch, both of those models give you patch or region level representation, but it would be nice to have a representation at the level of the slide. So single feature vector representing the entire slide. If you have that, of course, your learning problems on top of it would become easy. But also, we have to make sure that a single vector representing a gigapixel slide is sensitive enough of the morphology within the slide. So we started by looking at sort of these very obvious things like, well, we have all these morphologies. Can we use something like Gaussian mixture models to just come up with a slide level representation? And we did that and showed that this is possible. I mean, these are just some examples in survival outcome prediction. This was a CVPR study from 2023. More recently, we've expanded this into a much larger effort, and we call this model Titan. It's a slide level model, and it trains at 3 levels. The first level is at just the image level, where we use iBot self-supervised training, and we sort of brought in concepts that are typically used for large language models, like train short, test long, typically used to develop these large language models or multimodal large language models, where you're training on a shorter context and then testing on a larger context. So in this case, we are training on AK by AK and then testing at the entire slide level to get a slide level representation. But also by contrasting with 2 kinds of text information. So the first one is morphologic description that's generated using our generative model that I'll talk about in a second, but also by contrasting with pathology reports. And the contrastive learning approach helps us improve representation. So as these models are getting better, our benchmarks are also improving. So we don't show results here for basic subtyping or grading, and because it's saturated in performance for all of them. In this case, we're showing much more complex benchmarks, like 108 class rare disease prediction. But key results here is that you can look at the scaling laws, what happens when you increase the number of data points used for training. But you can also look at what happens with image contrasting. So how the representation improves, text contrasting, when you contrast with morphologic descriptions to improve the image representation. And of course, you can look at few shot performance on all of these different tasks as well by using very, very few labels, which really is very important in pathology, because a lot of very interesting questions that we want to answer, that what's the next patient that's best to enroll in a particular clinical trial, rare disease, all are effectively few-shot problems. We don't have lots of data available for them anyways. And then retrieval and similar slide search. So this is something that's inherently done in pathology for rare disease. So the typical workflow, unfortunately, is still that every major pathology department would have somebody who specializes in these kinds of rare diseases, and they would look at a case and try to compare it with an existing case or a case that existed a long time ago, and that comparative analysis would then lead to a diagnosis. And here we try to do it computationally because we have slide level representation. It's very easy to build a database, find what the most closest slide would be in feature space, and could help diagnose rare disease. And then we assess this on large amounts of internal cancer data as well as on external data. So if we just recap some of the story that I've told, that multiple instance learning is the most commonly used paradigm for processing pathology whole slide images into some kind of meaningful learning paradigm, and feature representation is important. And you can use self-supervision to improve feature representation, and then contrasting with additional modalities improves image representation as well. So just in light of that, there's a lot of other data in pathology that you could be contrasting with, right? So we show that you can contrast with text to improve representation, but there's also morphologic data, molecular data, and also immunohistochemistry, radiology, other kinds of data that you can contrast with to potentially improve image representation. So we tried to test this hypothesis in a number of the different studies. So this is from ICCV last year, where we contrast H&E images with IHC images and special stains. And by contrasting with IHC images and special stains, we effectively show that you can improve image representation both for the H&E and for the IHC, and we applied it to then a number of different downstream tasks. So if we can contrast with the immunohistochemistry, we can also contrast with molecular data, right? So contrasting with molecular data, and this study is from CVPR last year, where we're contrasting with molecular data to improve image representation. And what's important is that we found that contrasting with transcriptomics, this was a sort of a toy study just done on the TCGA to assess this hypothesis that contrasting with transcriptomics would do better than contrasting with, for example, text. So text is very subjective, but transcriptomics is much more objective. So if it's true that it relies on what's common across these multiple modalities to improve image representation, we should get a much richer slide level representation by contrasting with transcriptomics. And that's what we found. So for the 3 different cases that were studied in this small study, we found that few-shot performance improved substantially when contrasting with the molecular data. So this was first indication that this aspect should be scaled. So that's what we did in this study that we call Threads. And this is, we have a preprint for this, and this will be coming out soon. But we basically contrasted with all the molecular data that was available to us from the Dana-Farber Brigham profile cohort, as well as from MGH. And because this is contrasting and not fusion, we can have separate heads for each type of molecular data that is included. So the goal is not to fuse this information to get to a particular answer, but the goal is to contrast across this information such that to improve image representation. And we found that by doing that, we were able to improve performance in a number of very difficult tasks that are quite challenging, like treatment response prediction. Like here, there are all these different drugs, and we're predicting treatment response directly from the histology image. And for some of these, there's the drastic improvement when using this model that was trained by contrasting between histology and molecular data. So we're still scaling this with more data from other institutions and see how far we can go. We can go. Other people have also indicated to us that this is sort of a proof of concept that we can contrast against lots of modalities and improve representations for each one of those individual modalities. So everything that I've discussed so far is about improving image representation in pathology that can then be made more specific to lots of downstream tasks, answering specific questions. But if you have rich representation from these images, one obvious thing we can do with this is to build a co-pilot or a generative model that you can then use to answer questions. So what would we need to do to build a multimodal large language model that would cater to all of human pathology? You would basically need rich image representations, which we already have, using some of the foundation models that we had developed, but we also need representation, but we also need instructions. So the philosophy behind creating instructions is that pathology data is hierarchical, harbors information at many different scales. A good chatbot or a multimodal large language model should be able to answer questions at any level of abstraction, but we don't have that morphologic description readily available to us. The only thing we have is pathology reports corresponding the entire slide. So we needed to create lots of instructions. So we started to do that manually and managed to create quite a lot of instructions manually, but over time it became clear that this was sort of a very difficult task. So we made use of a lot of training materials that exist at Harvard hospitals and other institutions, other collaborators who contributed training materials. It's effectively using the same materials that are used to train residents and pathologists to train this multimodal large language model. And once we had the model trained, we could then go in and start asking questions about regions within the pathology image. In the beginning, we're targeting very fundamental and simple questions, and over time, this has become like a large effort to really scale this. We could also just couple a cell phone to a microscope. It's very common to do this in low resource settings. And pathologists often send their colleagues in more resource-rich settings images that are taken in this manner. So we can get responses right away. Some foundations that we do some work with are very interested in using this in lower source settings to get immediate answers, because it's typically quite difficult for them to track the patient back once they've left the city where the hospital is. They can't just send the report over their phone. So this is also of interest in lower source settings. But the real proof is in the assessment. So we did a lot of initial assessment that was based on multiple choice questions, comparing with more general models like GPT-4. And then we also asked a panel of 7 board certified pathologists to compare responses from this model versus other models and how well this approach does for diagnosis and morphologic description. So this has sort of been licensed to a startup, and they've taken it further, and they recently got FDA breakthrough device designation, and will be running a clinical trial to try to see if it can improve workflows for triage, automatic test ordering, and for report synthesis as well. The next thing I want to touch upon is, I guess everyone's talking about AI agents. So we have been trying to build some of these agents for computational pathology. And since then, it's been expanded to more of agents that would do any kind of analysis with tissues. But the idea is that, of course, AI agents do things for you, and everything that we're doing back in 2019, 2020 can be done by these agents more or less on their own. So the effective benefit would be that you can reduce the barrier for who could be developing these kinds of models. And quite often, people who work in domain-specific pathology or oncologists, they often have the right questions that they want to answer. So maybe they don't need to go to a computational person, someone like us, to run these experiments. Maybe they can run it on their own. Agentic AI is really ripe for computational biomedicine in general, because generative models have become quite good. So if you have a text prompt, you can easily prompt, you could easily generate a plan from that text prompt. So the current large language models from OpenAI and Anthropic are very good at doing this, and it can create a plan for you. And then the agentic aspect of this would be that you can then schedule and get access all those tools, and it would go run those experiments for you, and you would effectively be able to communicate across it. So we started building this agent. Initially, it was just an effort that was centered around pathology, and as the studies were sort of going through the review process, and we had requests that can we add other kinds of tissue analysis in there besides just looking at pathology images? Can we look at spatial transcriptomics? Can we look at radiology and other kinds of data? So that's what we have effectively focused on. And while we were building this, there was also lots of development that's happening. Coding is becoming from text prompts to effective code is becoming better and better. And then our PathChat model has also improved quite a lot, so we can do good morphologic descriptions. And then OpenAI also released deep research, so we can also go back and do more generic scientific research, and then come back with generated hypotheses. So if you put it all together, a lot of these experiments could run effectively on their own. This is just one example of it, where the user is giving it some metadata and saying that I have a cohort of responders versus non-responders, and can we look at what are the morphologic differences, and then maybe conduct some analysis on top of it. So the generative aspect of this is that it can make a plan, and the agentic aspect is that it can then go and execute that plan. So in this case, it's very simple, but it's a starting point where you can segment the tissue, patch it, extract features. Once those features are extracted, you can train a model on top of it. Once the model is trained, this video is sped up because the training process takes time. And once you have the model trained, you can look at the AUC for how separable the responders from non-responders are. So the AUC is about 0.855, maybe not clinically useful, but it does give an indication that there's a morphologic difference between the responders and non-responders. So then you might want to investigate what those differences are, and you can look at some form of interpretability, and you can look at these heat maps, which don't tell you too much, but it does give you an idea for what the model used in making its predictive determinations. And another thing we could do, because we have the generative model, we can ask the agent to effectively look at the highest attended regions in all of these slides in your test set, and give a morphologic description for what's common across this. And it does that, and it tells you, well, it's looking at inflammatory response, necrosis, fibrosis. And then maybe you want some more enhanced interpretability. You can say within your high attention regions, can you segment all the cells, extract handcrafted features, and give me differences between responders and non-responders in a more clear manner, and you can do that too. So there are other examples of this that we have out there, but we'll have more to say about this in the coming months. I also want to say a little bit about transitioning from 2D to 3D pathology. So pathology today is inherently two-dimensional. So human tissue is three-dimensional, and clinically and for other applications, we typically look at a really sampled view of the tissue. And for many disease indications, this works just fine. But for others, it has been shown that as you cut additional images from a tissue block, the diagnosis changes as more and more data is sort of observed from this piece of tissue. And over the past few years, there has been just an immense amount of effort devoted to making three-dimensional imaging easier. And this includes methods that could include serial sectioning of tissue and then registration, or using micro CT scanners, which is the relatively easy way of doing it, or using open-top light sheet microscopy, which gives you very high resolution, but it takes a little bit more time and requires tissue clearing. But there's no good computational pipelines that I can just directly use on three-dimensional data, and this data is very large and complex. Whole clad images are already, can be 100,000 by 100,000 pixels, and you add another dimension to it, it becomes very, very complex. And the other aspect is that if we are able to image this tissue in 3D, and pathologists are looking at this using just their eyes, it will be impossible to cater to the patient workload. And it's also difficult to discover from this data. For discovery, we're effectively interested in finding common morphologic features across large amounts of patients that correlate with either response or resistance to treatment, or just to separate them into distinct risk groups, which is also difficult to do manually. So we try to develop computational pipelines for three-dimensional pathology data. Of course, this data is just very, very computationally complex. Very, very large images. These are prostate cancer cases. They were imaged at Harvard using data from the Brigham and Women's Hospital using a micro CT scanner. But we also had another cohort that came from an open-top light sheet microscope from Jonathan Liu's group at the University of Washington. And we expanded multiple instance learning into a third dimension, which was just complex, and it requires a lot of computing resources to train models like this. But basically, we managed to scale this, and it follows a similar kind of a paradigm where we extract features, we aggregate, and then we pose the problem that we're trying to solve. This was a Cell article from last year where we managed to show that as you use more of the tissue region, you're able to separate patients into distinct risk groups in a much better way. And since then, we've been expanding this to other applications, and we'll have more to say about that in the coming months. But you're able to go back and look at what are the regions that are important within this large piece of tissue. Sorry, I'm trying to. Okay, so another question. So as we're doing this, there were multiple of our collaborators have asked us this question: that can we infer spatial transcriptomics in 3D now that we have lots of spatial transcriptomic data available, those that can be used for pre-training. So the basic idea is, and this idea is not unique to us. It's being explored by many other people. So we have tissue, which you can image using any kind of 3D imaging technique. In this case, we relied on micro CT scanning because it's a good trade-off between resolution and how quickly can you image these cases. And then do few two-dimensional spatial transcriptomic sections on this piece of tissue, followed by using a pre-trained model that might have been trained on lots of paired histology and spatial transcriptomic data. So it's learned that basic morphologic correlation, and then fine-tuning, in-patient fine-tuning on samples that actually came from within that piece of tissue to then infer spatial transcriptomics in 3D. And now this is, of course, it's we try to do a proof of concept of this, and it works to a degree, but we are trying to validate this further. We sort of used every architectural combination that we could, including using multiple sections, all kinds of different architectures on the machine learning side, as well as all kinds of different validation that we could do both on the pre-training end and on the post in-patient fine-tuning end to see if we can improve predictability, as well as validation by doing lots of multiple sections and spatial transcriptomics on it. This was limited to prostate cancer, and we have since expanded this, and we're also expanding this to use open-top light sheet microscopy, which gives us a much higher resolution view of this. We have a preprint for this, and the study is currently in review, but we will be making all the code and everything publicly available so people can experiment this with this sort of concept further, because it will take a while before it's actually perfect or gets even close to being perfect, if it ever will. But you can go in and, this is just fun images where you can go in and look at what it's effectively looking at and draw some conclusions from it, if possible. We also have efforts to look at more sort of treatment response related questions in 3 dimensions, and that's sort of ongoing work. We're obviously very interested in applying machine learning to real world settings, and one aspect of this is that how do these models perform when we individually analyze them on protected subgroups of patients. So we wanted to see if there's anything in the model architecture, what modeling choices can we make such that there will be minimal bias in these models. So the study from last year focused on analyzing some of this. So we trained some of these models on large public data like TCGA, other large public cohorts of pathology data that was available, and then assess these models on data from Brigham and MGH, which was stratified by protected subgroups, whether it was race, income groups, or whether the patient had insurance or not. And we found that there can be large disparities, but we also looked at what modeling choices could mitigate those disparities and found that feature representation is the most important component. But just feature representation, improving feature representation does not eliminate all of those disparities, and we're investigating why they exist. Sometimes it could be very obvious. An example would be that patients with socioeconomic disparities might just be diagnosed late, and there might be limited advanced cases within your test set. The last thing I want to touch upon is what are we working on next. So We have been building a model that uses lots of data from Brigham and MGH, is multimodal, uses all the modalities that we have available, and is also temporally aligned, such that we will get a single patient level representation for every patient that can be dynamically and temporally updated. So the idea would be that every event that occurs about the patient, whether it's a visit, it's a phone call, once it gets logged, either in electronic medical records, a radiology scan, or a pathology report, or a clinical laboratory test, would update a dynamic patient level embedding. So this effectively includes using self-supervised models for each individual modality to embed them, followed by a temporally aligned embedding. So with every visit, your embedding would effectively update, and you would have this sort of change across time recorded, which you can then apply to a host of different downstream applications like early diagnosis, treatment response, biomedical discovery, dynamic risk scores, clinical trial matching, population health, or just some examples. We are beginning to get some early results for this, and we'll have more to say about this in the coming months. I want to end by showing this poem from Judith Previtt. That I think she wrote something that is very relevant today, and this was written in 1979. She writes that optical illusions can deceive the subjective eye, but objective measurements and algorithms are assumed not to lie. It's often said that medicine could use such objectivity and thought that this justifies machine intelligence activity. Artificial intelligence is another craze that uses computers to cope with the diagnostic maze. Though the criteria for intelligence has never been resolved, paper after paper claims that the problem has already been solved. So I find it fascinating how relevant our words are today. So we really have ways to go before everything we do and others do in the field could actually make a true impact on the clinical and discovery side. It's beginning to show, but it will take more time. I want to thank all the PhD students, postdocs, and all the support staff who have contributed to all of this effort, as well as all the funding that we received to do some of this work. Thank you so much. I'll be happy.